So we're done with glycolysis, and glycolysis can be aerobic or anaerobic. That means glycolysis is going to run no matter what, whether your cell has plenty of oxygen, aerobic, or whether your cell doesn't have plenty of oxygen, anaerobic. Now, the fate of pyruvate, though, depends on if the cell is aerobic or anaerobic. If your muscle cell has enough oxygen in it, then your pyruvate is going to get turned into something called acetyl-CoA. And there's an enzyme called pyruvate dehydrogenase, and it's a redox reaction, and carbon dioxide is removed, and then you're putting, see, pyruvate is 3 carbons, the acetyl group is just 2 carbons, so you're going to put that acetyl group onto coenzyme A, and then coenzyme A is going to carry it into the Krebs cycle. All of this is going to go on in the matrix of the mitochondria. So under aerobic conditions, pyruvate gets turned into this acetyl-CoA plus carbon dioxide. And this is with oxygen. This is in a human muscle cell. This is what goes on if there's enough oxygen, okay? Remember, coenzyme A derived from pantothenic acid. The enzyme here is pyruvate dehydrogenase. Now, if there's not enough oxygen, you know, like when you have been exercising a lot and you start getting this muscle fatigue and muscle pain. So now you're in an anaerobic state. Pyruvate can't get turned into acetyl-CoA. Nothing can go into the mitochondria without enough oxygen. So instead, the pyruvate gets turned into lactate, and that's what causes that ache in your muscles. So this over here is without oxygen. So in a human muscle cell, either this or this, okay? Now, with enough oxygen, this is what's going to happen, and all of these things, then the acetyl-CoA goes into the Krebs cycle, and that's the topic of the biochemical energetics chapter that we'll do next. But if there's not enough oxygen, it gets turned into lactic acid. Now, the lactic acid is produced in the muscle, and then it goes to the liver where it goes through something called the Cori cycle, and then gets turned back into glucose, okay? So lactic acid can get turned into pyruvate and then glucose, and, you know, there's actually several different ways of doing this. I think I talked in the gluconeogenesis section, so we'll get to that in a little bit. Now, before I move on to this, in bacteria, they do this also. So under anaerobic conditions, so human muscle cells, but also bacteria will do this. They will turn, under anaerobic condition, they will turn pyruvate into lactate. And many, I don't know if you guys know how they make yogurt or how they make cheese, but usually non-pathogenic bacteria are involved, and they end up going through glycolysis and then turning the pyruvate into lactate, and then the lactate brings down the pH of the milk, and you get these precipitation of the milk proteins. So this is how the food industry uses glycolysis to make things like yogurt and cheese and stuff like that. Now, the ethanol, turning pyruvate into ethanol. Pyruvate is 3 carbon, ethanol is 2 carbons. So there's a carbon dioxide that's given off. This is actually a 2-step process. The pyruvate is first turned into acetaldehyde, and then the acetaldehyde is then turned into ethanol. It's reduced and turned into ethanol. By the way, all of these reactions are ways to replenish NAD. So you take all the NADHs that have been produced, and you can, this and this will replenish your NAD. This, no, this over here doesn't replenish NAD. This one actually uses NAD and then produces another NADH. So in this reaction, you end up also getting NADH. But these two, you are replenishing the NAD, meaning you're using NADH and you are actually getting some NAD back so that you can go through glycolysis again and again and again. The ethanol and carbon dioxide, this is yeast and plants. So we said humans and bacteria do the whole lactate thing without oxygen. If you don't have enough oxygen and you're a plant or yeast, you turn pyruvate into ethanol and carbon dioxide. What is ethanol? That's alcohol. All the alcoholic beverages, it's a process of glycolysis, okay? And I don't know if any of you guys have ever baked bread, but what do? You get yeast and you put a little bit of sugar and some warm water and you watch it do its thing and you start seeing bubbles. And the whole rising of the bread has to do with this carbon dioxide being produced, okay? So these are ways that the food industry uses glycolysis, specifically anaerobic glycolysis. We call it fermentation in order to make several different products. All right, so what you guys need to know is that the fate of pyruvate is several different fates. If there's enough oxygen, we're going to turn it into acetyl-CoA and carbon dioxide and NADH. Remember, everything is doubled. And then this guy goes into the Krebs cycle. If it's without enough oxygen in an anaerobic condition, so the term without oxygen is anaerobic. The term with oxygen is aerobic. So if you don't have enough oxygen, then if it's human muscle or if it's a bacteria, you turn pyruvate into lactate. If it's yeast or plants, we turn pyruvate into ethanol and carbon dioxide. Okay, so that is that. And now I think we're almost done. Okay, so let me give you the definitions of these terms, and then I think we are all set for carbohydrate metabolism, and then we can get into biochemical energetics, how your body makes the rest of the ATP, because so far we've only made two, remember? Just two. Okay, so the three terms I want to put up here are, and I don't know if they're all on your what you need to know list, but I just found these: glycogenesis and glycogen Lysis and Gluconeogenesis. So I already mentioned this when I mentioned the Krebs cycle, the way lactic acid, lactate, can be turned into glucose through the Krebs cycle. Okay, what we just covered was glycolysis, not to be confused with these things. Glycolysis is taking glucose, breaking it down into two pyruvates, producing two ATPs, two NADHs. Okay, that's glycolysis. Glycogenesis, what do you think that word is? It's glycogen and then nesis. So basically, it's the synthesis of glycogen. That's what glycogenesis is. So it's taking a bunch of glucoses, bonding them together, and synthesis of glycogen. And where is glycogen synthesized? We synthesize it in the liver and in the muscle. So all the extra glucose, it's a cycle. It's actually several steps, a pathway to do this. A uridine diphosphate is used. You can, look it up in the book about the details of it. You don't need to worry about the details. You just need to know the term, what it means. The other term is glycogen lysis or glycogenolysis. What does that mean? It is the lysis of glycogen. Let's say your blood glucose levels goes down. You are starving. You forgot to eat breakfast. You haven't been eating. So your glycogen, or you started to do a marathon, your glycogen has to be broken down and glucose goes into the bloodstream to go feed your brain and whatever cells need the glucose. So glycogenolysis is the breakdown of glycogen. Okay, the hormone that is involved with this, we mentioned it before, it's glucagon, not to be confused with glycogen. And then the gluconeogenesis, what that means is the synthesis of glucose from non-carbohydrate material. So it's not like you're starting off with a glycogen and turning it into glucose. This is what this is. Glycogen lysis is breaking down of glycogen and you end up with glucose. But gluconeogenesis is synthesis of glucose from non-carbohydrate material. So the Krebs cycle allows lactate to get turned into glucose. So that's one type of gluconeogenesis. Another type of gluconeogenesis is you can take that glycerol from the triglycerides and you can turn that into glucose. And then there are some amino acids, we call them glucogenic amino acids, meaning that those amino acids can be used to make some carbohydrate. And those amino acids we'll get into later on in the protein metabolism chapter, but we can write amino acids can be turned into glucose. So basically the term gluconeogenesis just means the synthesis of glucose from non-carbohydrate material. Okay, we are done with carbohydrate metabolism. I hope I've covered everything on our what you need to know list that I just dropped on the ground. All right, good. And in the next video, we will start with biochemical energetics. Okay.